this can be very energy intensive as all air brought in from outside has to be heated or cooled to match the building's temperature, costing a lot of money. Another option is filtration, although you often have to replace filters so often that it's neither cost effective nor efficient. We think a better solution is biowalls. Biowalls are vertical structures covered in plants which aim to naturally filter air and decrease VOCs. Additionally, studies have proved that added green space in the workplace can improve worker happiness and productivity, which gives biowalls an extra benefit. Though biowalls are such a great solution, unfortunately, most existing systems are often more aesthetic than functional, and that's exactly the issue we'll be targeting. There are two main types of biowalls. The first is an active biowall, where the wall hooks directly into the HVAC system of the building, pulling air from the room back through the wall and circulating it back through the rest of the building. This means that way more air comes into contact with the biowall, making it much more effective at degrading VOCs. However, the flow of air over plant roots is not natural and makes it really difficult for plants to survive. This wastes a lot of water as they need to be super hydrated to survive and creates lower plant density on the wall. Another option is passive biowalls, where the wall does not hook into the building's HVAC system, and the plants simply sit on the wall. This allows plants to be a lot healthier, and it requires less water to keep them alive. However, because they only come into contact with the air directly around them, they're not nearly as effective at degrading VOCs, and that's the gap we're trying to bridge with research. Scientists have known for a while that plants have the ability to degrade VOCs. However, for a long time, researchers thought it occurred in the leaves. Recent research at Drexel University showed that the degradation actually doesn't happen in the plants at all, but in the microbial communities on the plant's roots. Hypomicrobium is a genus of bacteria that's been specifically shown to degrade VOCs, and that's the genus we'll be looking at in our research. We have two research questions. First, what is the relationship between plant species and the concentration of hypomicrobium bacteria on the roots? And second, how well will a new passive biowall, incorporating a VOC dissolution system into the irrigation reservoir, improve filtration abilities? We hypothesize that one or more plant species will encourage more hypomicrobium growth, indicating a better choice for biowall use. Additionally, we hope that a new passive biowall with a VOC dissolution system will ameliorate more VOCs than a traditional passive biowall. Now I'm going to pass it off to Morgan to start to tell you how we'll answer our research questions.
So our methodology is split into three phases. The first two, plant and bubbler research, are happening alongside each other, and they'll come together at the end for our third stage, biowall system research. The whole concept behind our plant research is to answer that first research question and determine if plant species affects the concentrations of hypomicrobium on the plant roots. In order to quantify the hypomicrobium concentration, we'll be using quantitative polymerase chain reaction, or qPCR. An additional benefit of our plant research is that we'll be able to confirm previous research by Drexel University correlating VOC exposure to hypomicrobium concentrations. In order to monitor these VOC levels, we'll be using a photoionization detector, or a PID. So we have two steps for our plant research. The first is to test local passive biowalls for hypomicrobium, kind of to get an idea of where we're starting at, and also to test out our methodology. Our second step is to expose the plants to VOCs in aeroponic chambers. So these aeroponic chambers are basically boxes. We'll have the plants on top with only the roots hanging down, and we'll be spraying water, nutrients, and most importantly, our VOCs onto these roots, all the while monitoring the VOC levels and hypomicrobium concentrations. So far, in our plant research, we've taken the samples from local biowalls and used them to confirm our filtration methodology. Out of necessity, we did a two-step filtration. The first step is to remove large particulates and the roots, and the second step is to actually isolate the bacteria. From there, we've extracted the DNA, but we have to wait on qPCR because we just finished culturing our qPCR standards. So the way qPCR works is it fluorescently tags and amplifies the target DNA. In our case, our target is hypomicrobium, so our standard is hypomicrobium D19Ps. In order to get unknown concentration from your qPCR output, we need a standard curve of known concentration and their respective qPCR outputs. We've also set up our aeroponic chambers. So a big problem we faced here is that we can only afford one PID. So we only have one way to measure these VOCs, but we have 8 aeroponic chambers. In order to get around this, we set up a system of tubing so that when we want to go measure the VOCs, all we have to do is switch between air streams and we'll get all 8 measurements in one go. This leads me into our next step for plant research, which is doing the actual aeroponic testing. So like I said earlier, we have 8 aeroponic chambers. 4 will be controlled with no VOCs, and 4 will be experimental with the VOCs. This goes along with our 4 plant species, Asplenium nidus, Philodendron roja-congo, Chlorophyllum comosum, and Colloquia burlomars. We chose these 4 species for a variety of reasons, one of which being that they're different genuses, so they may have different interactions with bacteria on their roots, and the second of which we know they can live in an indoor setting, which is very important because indoor light is a much lower intensity than outside. What we're specifically looking for during aeroponic testing is first, how long it takes for the bacterial community compositions to adapt to VOC exposure. When Drexel did their research, they took a sample at 0 days and then a sample at 40 days, and noticed that in between, the bacterial communities shifted in response to the VOCs. What we'll be doing is taking samples more periodically to better gauge the shift. And once hypomicrobian concentrations level out, we'll be seeing how long it takes for the bacteria to remediate a set amount of VOCs. The information from these trials will directly answer our first research question and identify which plant species host the highest amount of hypomicrobium. Now that I've talked about plant research, Michelle will tell you about our bubbler research and how we plan on pulling everything together.
The next part of our research is what we've termed the bubbler phase because it centers around designing a mechanical system to dissolve VOCs into water by bubbling air into the system's irrigation reservoir. The purpose of this bubbler system is to transport VOCs to the roots of the plants in the biowall through the water, thereby improving filtration capabilities of the passive biowall without compromising plant health. The bubbler design consists of 2 main components, the pump, which introduces air into the water, and the water column, which provides sufficient time for the VOCs of this newly introduced air to dissolve into the water. The first step is to design an initial bubbler system and perform gas chromatography tests to ensure that the VOCs are indeed being introduced into the water. Then, once we've confirmed the functionality of our bubbler system, we'll introduce the mechanics into a traditional passive biowall design. Our original approach to designing this system was to calculate the best pump and water column combination to optimize the amount of VOCs we could saturate the water with. However, once we started this progress, we realized the calculations we needed to perform would have made too many assumptions about water flow, VOC diffusion rates, and air bubble distortions, thereby invalidating any results we would have gotten. Our revised plan is to vary pump power and water column height to experimentally determine the best combination. We'll then use gas chromatography to ensure that our chosen combination for the bubbler system is dissolving the VOCs into the water properly, and then integrate this system into a prototype biowall. Working with Michael Furbish, CEO and founder of Furbish, a green systems manufacturing company, we've been able to design our prototype walls to closely reflect current passive systems. For our prototype walls, we'll be using plants with the highest hypomicrobial concentrations based on results from our plant research phase earlier in the project. The basic design of our prototype walls consists of 2 felt layers at the front of the wall that are stapled together to form pockets where we can house the roots of the plants. These felt layers also support the plants on the wall surface and eliminate any need for soil. Behind these felt layers, we'll have a PVC board as well as a waterproof plywood backing. At the bottom of the biowall is a water basin to collect any excess water and recycle it back into the irrigation reservoir, which doubles as our water column. Attached to the water column is a pump that feeds 2 drip irrigation lines at the top of the biowall. And for our modified biowall, that including the bubbler system, we'll have the bubbler attached to the water column as well. Now the final step is to analyze our biowall systems. One wall will be our modified passive wall. That contains bubbler mechanics. And we'll also have a control, which is just a traditional passive system. We'll compare performance in 3 ways. PIV readings will monitor air VOC levels. Gas chromatography tests will monitor the water VOC levels. And qPCR conducted on root samples taken from each biowall will monitor the hypomicrobial concentrations. We'll compare the modified system against Drexel's active biowall to see how a passive system with our introduced bubbler mechanics competes against an active system. Now Jackie will talk about our team composition, challenges we faced, and our advice for the freshmen.
And
Morgan and Michelle explained our methodology kind of in two distinct sections, and that's exactly how we approached our team composition, by dividing into two groups. Our first sub-team deals with plants and bacteria, approaching the project from a biological perspective. The second sub-team works with the bubbler and wall design, looking at things more from an engineering perspective. We hope that this strategy will allow us to answer both of our research questions in the time allotted. Though we're only a junior team, we're already really excited about the potential applications for the results of our research. We someday hope for biowalls in more corporate and academic buildings to increase productivity for employees and students. We hope for smaller biowalls in private residences to further improve human health. And most exciting, we have a potential application coming up right here on campus. Last year, there was a proposal approved to put a biowall right here in McKelvin Library, and we've been in touch with the designers ever since. The project was supposed to be completed right about now, but fortunately for us, it was pushed back about a year, giving us plenty of time to collect data and better inform their project. But we've had a lot of success in the last year. We've also definitely faced some challenges, the largest of which has probably been timing. We faced some issues with ordering, with some confusion about placing orders, some back-ordered shipments, and that's definitely cost us some time. Additionally, we've learned that things are a lot more difficult in the lab than they seem on paper, so mastering our lab procedures has taken a little bit of extra time. There are some pretty big anticipated challenges coming up. Next semester, 3 of our 8 team members will be studying abroad. Fortunately, it's something we've known about for a long time, and we've been able to plan accordingly. Additionally, we are a little concerned about building our biowalls. The company we've been working with has been perfecting their techniques for about a decade, and we've only been at this for about a year. So hopefully with their guidance, we'll be able to create healthy and effective biowalls. We do have some advice for the freshmen. First of all, be flexible. Sometimes your interests and expertises may change, and you shouldn't be afraid to change with them. Additionally, be aware of what your teammates are going through. Everyone has difficult days, difficult weeks, even difficult semesters, and being understanding of that will only make you a better teammate. Lastly, something that's really helped our team, make sure you apply for funding early. We looked right here on campus, and in our very first semester as a team, we were really successful with LaunchUMD and the Sustainability Fund, allowing us to complete our project. Additionally, as you apply for grants, make sure that you're understanding your audience and adapting to their interests and knowledge, as that really pays off at the end. We want to extend some huge thank yous, first of all, to Dr. Andrzejewski and Dr. Stephen Cohen, our amazing mentors, along with Dr. Svetlana Bekacheva, our librarian. We also had a ton of help from different experts, both here on campus and over at Furbish Company, and we owe them a ton of thanks. And finally, financially, we owe a big thank you to the UMD Sustainability Office and everyone that donated to our LaunchUMD campaign. And of course, thank you to the Dennis Point staff. Does anyone have any questions?